Okay, ladies and gentlemen, thank you for your attendance. I'm Don Lindbergh, Director of National Library of Medicine, and in this case I'm welcoming you, and partly on behalf of NLM and partly on behalf of Medical Library Association, who join annually for a Leiter lecture. In this particular case, we regret that Joe Leiter can't be with us. He's in town, but seeing a doctor, but only temporarily. As you perhaps know, the series started in '83. I think my colleague Marty Cummings gave the first such lecture in 1984, and there have been a string of splendid presentations by outstanding scientists, capped by Dr. Collins, I might say, who will address us on the topic Genomics, Medicine, and Society. I should tell you a little, very briefly, a bit about him. He became director of the National Human Genome Research Institute in '93, replacing his successor, Jim Watson, who is a perfectly wonderful person to have as your predecessor, I might say. And Francis has done a splendid job in that position. His education includes a undergraduate degree in chemistry from UVA, happy to say, a lovely institution to the south, and a Ph.D. in phys chem at Yale. And somehow or another that got him pretty launched in science, but he decided that he liked to have a little medicine background too, and took an MD at University of North Carolina, where he also took the opportunity to learn clinical medicine and become chief resident in medicine. That is the point on which one knows the most will ever know. At least he thinks that. But I had the personal opportunity to confirm, with having fallen ill on a field trip, that Dr. Collins still has the clinician's touch and still is a good doctor. And I'm alive. I survived his treatments. His contributions in genetics are truly too numerous to detail at all, but I should say that while he was still at Michigan in '89, he discovered the cystic fibrosis gene, in 1990 neurofibromatosis, subsequently here ataxia telangiectasia, and he's made major contributions to the understanding of the genetics of Huntington's disease and breast cancer as well. As I say, the scientific publications are all important and all marvelous and too numerous to name. I do want to say that Francis has always been very attuned to public policy issues that stem from the Human Genome Project and the work of his institute, since it clearly redefines medicine every day for us. And so he has drawn attention both here and in the public and in the Congress to the importance of our earnest desire, yet unfulfilled, for proper national legislation assuring that we won't have genetic discrimination based on genetic testing, and of course to assure medical data privacy as well. We await both of these bills anxiously. One of the technical contributions Dr. Collins made to the field is a technique called positional cloning, and I sort of sensed that, like many innovations, it wasn't greeted with 100% applause on all sides, and I decided that must be right when I looked over his CV and found that in 1995 he has a paper called Positional Cloning Moves from Perditional to Traditional. So I gather you must have won that argument in the long run. Without delaying further, I wish to bring to the podium my colleague and good friend, Francis Collins.
Thank you, Don, for a very kind introduction. It's always a pleasure to be introduced by somebody who's both a colleague and a friend, and Don has been both of those in the 9 years since I've been here, and it was a particular pleasure and honor to be invited by the NLM and the MLA to come and give this lecture, the Joseph Leiter Lecture, especially noticing the distinguished list of speakers who have preceded me at this podium. In thinking about how to make a presentation to this particular group, I thought I would try to step back a bit from the minutiae of how many base pairs got sequenced yesterday or the day before, and instead to paint a broader picture of where we've come from in the Genome Project over this last dozen years, but particularly to take the occasion to talk a bit about where we're going. Because while I think there is a general sense that we have crossed a historic bridge with now the letters of the Human Instruction Book largely available on the internet for everybody to look at, we still have a very large amount of work to do if all of this information is going to fulfill the promise that was the reason for doing the project in the first place, which is namely the benefit of understanding the genome for human health. So I'll say a bit about that. That's the medicine part of the title. I also, though, think that in a gathering of this sort, that we ought to think in an even larger way about the consequences of this rapid acceleration in the pace of our understanding about human biology and genetics, and talk a bit about some of the challenges that face us as a society in making sure that these advances are used to benefit humankind and not used to injure people. And as Don mentioned in the introduction, there are some issues here that require close attention and even legislation, and I'll point out a few of those near the end. So that's my intention, and you can decide if we get there or not. I was a little mixed up when I came to give this presentation. I went to the wrong auditorium over at Natcher, thinking that that's where the lecture was going to be given, because at some earlier point that was a possibility. I got there and discovered that there's a lecture there on pain, depression, and fear. I thought for a moment that maybe that was somebody's notion of what I was talking about, and yet I hope by the time we get to the end of this and go out for a nice reception that you won't decide that that really was the title I should have used. Well, okay. So let's get into this particular topic, and of course we have to start with a quick reminder of what this molecule is that the fuss is all about. It is almost now 50 years, it will be next April, since Watson and Crick published their seminal one-page paper in Nature, which detailed the double helical structure of DNA and laid out the way in which this biological molecule carries information, a fashion which, of course, at that point in 1953, was considered rather radical and surprising, but in these days where so much of our thinking is built upon the digital understanding of how information is stored in computers, this clearly resonates with that perspective of an elegant and efficient way to store information and even to pass it along by making copies. And of course, Watson and Crick pointed out that the way in which information in all living organisms is carried is by the simple series of these letters A, C, G, and T, standing for the bases of the DNA helix. And it still amazes most of us who contemplate this, that this system is adequate to carry enough information to specify a human being in those 3 billion letters of the human genome, but clearly the system works. It's also the case, though, that misspellings can occur in such a large code, and it's those misspellings that are at the heart of risk for various illnesses and therefore of major importance in our effort to understand how the genome works. So here we are talking about the Medical Library Association. I guess I often use the metaphor of DNA as an instruction book, or maybe it ought to be a whole set of volumes because there's an awful lot of information here. But I think it is probably our best metaphor. It's also sometimes called a blueprint. I don't think that works quite as well. It is, after all, a linear, one-dimensional code which can be understood in the same way that you would trying to understand a text, except that, of course, this one is written in a very strange language, a language that we have yet to really understand in anything except the most rudimentary way. And I think that tells you that a lot of what has to happen next is the process of translation. We use translation to talk about moving basic science to the clinic. In that regard, that's sort of an interesting alternative meaning of the word. Here we need to translate the DNA instructions to understand what they mean, and that also will move us from the bench to the bedside. First, let's be sure that the consequences of our understanding of this instruction book are fully realized, because I think some people still consider that the science of genetics, when applied to human health, is relegated to the study of relatively rare diseases that occur in Mendelian or chromosomal fashion. And in fact, it is clear and has been known for decades that that is far too narrow a view, that virtually any disease that you study, if you study it hard enough, you will see there are hereditary factors that play into it. Perhaps there are some exceptions in cases of trauma, but I would challenge you to come up with other conditions where heredity does not play a role in predisposition, and some would argue that that applies to some cases of trauma as well. Now, in fact, one way to think about this is to contemplate what runs in your own family, because most of us have a family history of something that has occurred at a higher frequency than it seems to in the general population. That is a reasonable hint as to what might be in your particular DNA instruction book, because I'm sorry to break the news if this is not something you're already aware of, but there aren't any perfect specimens here in this room or anywhere else. We're all walking around with a certain number of these glitches that place us at risk for future illness. Now, don't get me wrong. I am not by that statement implying that all illness is hardwired into your DNA and there's nothing you can do about it. In fact, many of those predispositions interact very heavily with environmental triggers. And for most common diseases like diabetes or heart disease or cancer, it is that interaction between a series of genes and environmental exposures that result in health or illness. But by studying the genome, by uncovering the hereditary contributions, we will probably have a more powerful platform to understand what the environment has to contribute as well. And so one should not think of this as a circumstance where you have to sort of be a believer in heredity or a believer in environment. We should be believers in both of those things, and we should take every opportunity that science offers to try to unravel those complicated relationships. And right now, the study of the genome is perhaps one of the best opportunities we've ever had to begin to make sense out of that complicated set of contributors to disease. The Genome Project is now 12 years old, having started in 1990 under the able leadership here at NIH of Jim Watson, as Don mentioned. But it was an international project as well, with contribution by a number of countries, and particularly when it came to sequencing the human genome, our colleagues in Japan, China, France, Germany, and particularly England were major players in getting this work done. And it has been my great privilege over the course of the last 9 years to try to lead that team effort, a team effort that has involved hundreds, in fact thousands of scientists in many laboratories, all banded together to try to achieve this common goal of unraveling the letters of the code and beginning to try to understand what they mean. It was not possible at the beginning, however, to just start sequencing human DNA. This timeline shows you some of the things that happened in the first 10 years of the Genome Project, and many of those milestones that you see marked by yellow arrows are not even about human DNA. They are about other organisms that were quite wisely included in this project because of the opportunities they provide to understand fundamental principles of biology that are going to be necessary for us to understand if we're going to ever figure out how human biology works. So I won't go into what each of these arrows represent, only to say that each one of these was a moment at which a data set of some sort was produced that was pretty valuable. And I'm happy to say that each one of these was achieved on or ahead of the schedule that was originally set for this project back in 1990 when it began, building upon recommendations of a very distinguished panel of the National Research Council, chaired by Bruce Alberts, that got the whole process going. The sequencing of the human genome, the reading out of those 3 billion letters, did not begin until 1996, and even then on a pilot scale, because we were not sure just how exactly to accomplish a truly prodigious amount of work, which would require us going from sequencing rates that were already impressive, but probably 2 orders of magnitude below what they needed to become in order to achieve the sequencing of the human genome in a manageable time period. Between 96 and 99, those pilot projects carried out in about a dozen centers in the U.S. and elsewhere went forward. And by 99, it was clear that many of the hurdles had been crossed, that automation had been developed to take care of some of the thorniest steps that were previously too expensive or too error-prone, and it was time to scale up and actually go for broke. That meant assembling, again, a remarkable team of individuals, and this is a photograph taken in 99 at one of the many gatherings of the Genome Center directors who were charged with doing this work. This particular picture taken in England at Hingston Hall, where the Sanger Center is located. And these folks basically divided up the genome into territory, and each center took a responsibility for sequencing a particular region, and the size of the region was chosen to be proportional to the capacity of the center. And everybody had to agree to certain standards of quality. We wanted to do this at high quality. And also had to agree that if they began to fall behind in production, that there would have to be some rearrangement of territory, because we didn't want to have part of the genome sort of left there unattended to for a prolonged period of time. And that was at times a little tense because people had to actually admit exactly what was going on. But we were greatly aided in that by a decision made by this group back in 1996, which was that all the data coming out of sequencing the human genome would be placed on the internet, made available through GenBank, this wonderful sequence resource run right here at the NCBI, every 24 hours. And that was done because of the very strong feeling of this group that this data would really only be of public benefit if it was immediately accessible to anybody who had a good idea about how to use it. And so no patents were filed on any of this sequence information, and all of it was, in fact, immediately placed the same day it came off the sequencing machine onto the internet databases where anybody who wanted to look at it could do so. And I think when history looks back on this whole process, and particularly perhaps on the process of a competition that subsequently arose between this public effort and a private sector effort, the notion of free and unfettered accessibility will be considered as a cardinal feature of the public effort, and one which in the long term was probably highly beneficial to the progress of science. I would point you to a very interesting essay on that particular aspect of the project, as well as sort of an insider's view about how this all went, that is just out in the current issue of the Journal of Molecular Biology and authored by Maynard Olson, a very distinguished genome scientist and actually a member of the original NRC panel that put together the plans for the Genome Project, and who subsequently became a major leader in the effort, including being a leader of one of the genome centers. And if you want to get a view of how all of this played out, and a very interesting and entertaining one, you might go and have a look at Maynard's just published article. You might also be interested in reading a book by John Sulston, one of the major architects of the effort. That's John up there in the beard. He's now Sir John Sulston, and led the effort at the Sanger Center. And his book, The Common Thread, was published in England a couple of months ago and is coming out in the U.S. edition in about a couple of months, and also makes a pretty interesting read. One of these days I hope to write my own book about this, but I think I'll have to retire from federal service first. Otherwise, somebody will have to clear the whole thing, and who knows what would happen then. So these people were charged with doing something pretty remarkable. We had to collectively achieve a sequencing capacity of 1,000 base pairs a second, 7 days a week, 24 hours a day, in order to sequence the human genome in this prospected 2-year period, at the end of which we hope to have a draft sequence covering the whole thing. And when you consider that when I was a postdoc, sequencing 1,000 base pairs was almost enough to get you a Ph.D., the idea that we're going to do that every second in a sustained way is truly a dramatic step forward in terms of throughput. And of course, this all had to be done at an affordable cost as well. That led to the construction of very impressive sequencing centers like this one at the Whitehead Institute in Boston, where you will see an awful lot of equipment, in this case conveyor belts upon which sequencing templates are moving from left to right. You'll also notice that there aren't very many people in this particular laboratory because the work is essentially all automated, and the people are all upstairs sitting in front of the computer screens having a glorious good time analyzing the data as it comes out, which is in fact a very interesting part of this, and perhaps the one that was the most demanding in terms of creativity, intellect, and simple body count, because if you really wanted to analyze this data, you needed an awful lot of input. And I think one of the lessons we've learned early on is that the computational side of genomics is going to be perhaps the most interesting and perhaps the most demanding. And so places like the NCBI right here with their wonderful collection of intellects and capabilities have played an absolutely central role in getting us as far as we've gotten in genomics, and will be even more essential in the future. With all of this effort, it was in fact possible to generate a draft sequence of the human genome by June of 2000. And there was a big press event there in the White House where Craig Venter, who led the private sector effort at Celera, and myself got to stand with the president in the East Room and link up with a satellite hookup to number 10 Downing Street and announce to the world that we now had a 90% complete draft of our own instruction book. And that was a very remarkable day, to be sure. But I think scientifically, what one puts more weight upon is the publication of such efforts after appropriate peer review. And so I think for our money, the real milestone was the publication about a year ago in Nature of this description of the sequence and analysis of the human genome. This is the longest paper that Nature has ever published, so it's not sort of a simple read on a coffee break, but it still, I think, is worth having a look at if you've got a day or so. And hopefully, when we look back in 10 years at this particular paper, we won't be too red in the face. I'm sure there will be some aspects of the analysis that we will subsequently learn were naive or misguided. But I think for the most part, this opportunity to look at what our own instruction book had to tell us for the very first time was indeed exciting. That same week, Science published the Solera Genomics Corporation's review of the sequence of the human genome. And interestingly, as far as the interpretation of what the genome contains, there are great similarities between these two perspectives. The strategy that was used for doing the sequencing was different. As it turned out, since the public data was, after all, public, about 60% of the data in the Science paper was actually the same data, the data produced by the public consortium. So it's not surprising, therefore, that the sequence itself, if you looked at the A's, G's, and T's, showed great similarity. Of course, it should show great similarity since it is, after all, the human genome. And outside of that 0.1% where people differ, the rest of it ought to look the same regardless of who's doing the work. I should say that this cover of Nature, and I think probably the same applies to the cover of Science, both were attempting to be symbolic here in the picture painted of what was going on. In the case of the Nature cover, you will recognize the double helix image of DNA, but it's made up as a mosaic of the faces of individuals from all over the world of every imaginable form of dress, ethnicity, race, and gender. And that was an intentional message we were trying to send, that this wasn't just about some chemical called DNA. This is really about the human species, and it certainly is a moment in the history of our species that scientifically is rather profound, and one that I think we will look back on over decades as having been a very major transition in our own understanding of ourselves. Of course, we couldn't resist, having decided to make the cover this way, to play a couple of games and try to hide in this set of images some famous people because, you know, you got to have a little fun. And Where's Waldo is always a game that people have enjoyed. So on this particular cover, there is included a picture of Watson and Crick in their famous pose from 1953, admiring their model of the double helix. And I've discovered it takes the average student about 10 minutes to find it. And that seems sort of a waste of everybody's time, so I'm going to tell you where it is so that you won't go to that trouble. It's right there. And there they are. Actually, there was one other scientist who it seemed deserved to be put on the cover because of his role in founding the whole field, and that, of course, is Gregor Mendel. So Mendel's photograph was duly identified and included in the image. But unfortunately, when Nature prepared the cover, they trimmed the margins quite a bit. And so Mendel's forehead is the only part that you can still see. It is there, but that's all of him that made the cover. I guess it's an appropriate part. He was a pretty smart guy, but you'd have a hard time identifying the rest of him. Well, what did we learn about this? I could go on at some length about the surprises that came out of this analysis, but I'll just mention a few. I guess one of the things we all worried about is that there wouldn't be any surprises. It would all be just sort of like what you expected. And that would be sort of a bummer after all of this hard work. We needn't have worried. Perhaps the most dramatic finding, and the one which still seems to be holding up now after many months of reanalysis, is that the number of genes, by which I mean the number of DNA fragments that code for protein in the human genome, is much fewer than expected, probably in the neighborhood of 30,000. Whereas we had anticipated, and all the textbooks had been saying for some years, that there were going to be about 100,000. It is remarkable that we get by with such a small gene count, especially when you consider that other organisms that we don't hold in such high esteem seem to have gene counts dangerously close to our own. Take, for instance, the roundworm has about 19,000 genes, only has 959 cells and wriggles around in the dirt, and yet it has a gene count that's about 60% of ours. Even more disturbing, when rice was sequenced a couple of months ago, the gene count that came out of that analysis stands at about 55,000. So, yeah, have more respect for dinner tonight. There's a lot going on there biologically that we haven't had a full grasp of. Now, I still think that probably we can claim greater biological complexity than roundworms or rice. So there must be perhaps some reason for that. One part of that, and again this came out of the analysis, is that on the average a human gene makes about 3 proteins using this process of alternative splicing. And other organisms of a so-called lower sort, although they probably don't appreciate being called lower organisms, do this somewhat less abundantly. But that's not the only explanation. I think it's also interesting when you look at the architecture of mammalian proteins. They tend to be more complex than their homologs in other species like flies or worms, which may add to the ability of proteins to do some multitasking that allows us to get away with such a small list. But it is a profoundly small list. The idea that you can build a human being from a one-celled embryo to where you are now, sitting here this afternoon, with a mere 30,000 genes is really quite stunning. Now let me say, a very interesting aspect of all this is the fact that when I say genes, I'm talking about genes that code for protein. And increasingly, and there's a whole issue of Nature, a special summary or a supplement that goes into that that's just out last week, it seems that there may be a whole universe there of genes that code for structural RNAs, some of them very short and therefore very hard to detect, that may play a profound role in regulation, and that we've systematically not had a very good way of identifying those. So there's a whole field that's building up here to try to identify a larger set. And it would not surprise me if there are thousands, or maybe even tens of thousands, of those very short RNA encoding genes that have previously mostly escaped people's notice. And perhaps that will partly account for this big discrepancy in gene count and biological complexity. Another thing we learned by studying the Y chromosome and comparing it to the rest of the genome is that males make mistakes passing their DNA on to their children more often than females do. Some of you are not surprised by that statement, but we now have data to prove it. In fact, because you can discern the molecular clock by looking at the Y chromosome and compare it to other chromosomes, and the Y only travels from father to son, so it only goes through male meiosis, you can get a fix on the rate of mutations, and in fact, it's about 2 to 1. So that means that males are responsible for 2/3 of genetic disease. It has to start somewhere. But they can also take credit for 2/3 of evolutionary progress. You can decide which of those appeals to you more. Another fascinating aspect of the analysis, and one which occupies quite a number of pages in that Nature paper, is the stuff that's been called junk, the repeats. And at least one category of those repeats, the famous ALU element, which is repeated some 500,000 times in your genome and mine, turns out, when you have the whole set in front of you, to have the earmarks of something that evolution has wanted to hold on to, in that those ALU repeats are more abundant in the areas that are gene-rich than in the areas that are gene-poor, at least if you look at the older ones where evolution has had a chance to act. And that is a very surprising finding, because you would have thought just the reverse, that such repeats would be getting in the way if you landed in the midst of a gene-rich area, and that therefore they would be lost fairly rapidly. And instead, they seem to be selectively retained. We do not understand what that selective pressure is that causes ALU elements to be selectively retained in those regions, but there must be something, and that's a very interesting research question at the present time. Well, let me say that all of this information is now available to the scientific community, and particularly so through the resources provided by NCBI. So I have to put a plug in here for my good colleagues, David Lippman and all of his wonderful team, in the way in which they have made resources available so that anybody who wants to go and mine these databases for nuggets of wisdom can do so. And I suspect many of you in this auditorium are involved in operating these databases, and my hat is off to you, because they are wonderful resources that are used tens of thousands of times an hour by people all over the world. And I think much of the progress that's occurred as a consequence of the Genome Project has been made possible because of that immediate access, and access in a fashion that provides not only the data, but interesting views of the data and powerful tools to enable people to query the data. This, of course, is the home page for the NCBI. It prominently features the draft Human Genome right here. And, of course, if you click on that, it will go and show you some other entrees to looking at the genome and its sequence. If you want to then go at the map viewer and actually look at a part of the genome, here is part of chromosome 20, actually all of chromosome 20 in this particular zoomed out view, that would allow you to go all the way from the chromosomal view down to the sequence in a very powerful fashion that this map viewer provides. I might also say the assembly of the human genome, because this is a draft, has not been perfect, The most recent assembly, so-called Build 30, that Greg Schuler and his colleagues have just put together, is a substantial leap forward in terms of correcting some of the inversions and rearrangements that were present in previous versions, because we have a lot more data now on how to correct those things. And Build 30, I believe, is about to go up on the NCBI site in the next week or so. And if you're one of those who's using the human sequence for various research questions, watch for Build 30. You'll like it a lot. It will probably clear up some of the areas that are potentially causing you trouble if you're interested in long-range contiguity. I might say also that I like very much, a little bit more of advertisement for NCBI here, this coffee break feature, which is up on their website, which has various exercises that one can do in the way of learning more about particular aspects of the genome. Here is this issue of small RNAs that I was just mentioning that may be involved in regulation of development and could, in fact, be a profoundly important part of the genome that we haven't understood very well. And, of course, there's also this opportunity to tour the draft sequence and find out the most powerful ways to use it, which can be a little daunting to a beginning investigator who hasn't already been through all of these websites over and over again. Another very important website and database operated here at NCBI is the Online Mendelian Inheritance in Man, which I, as a physician geneticist, use several times a week to try to identify particular features of Mendelian disorders. And this is extremely well curated and updated, so I often can find on this references to articles that haven't yet appeared in my inbox. Another place that clinicians increasingly are going to look is this particular site of gene tests and gene clinics, also now operated here as a joint project of the NLM and the NHGRI, and which contains an increasingly large amount of information for the busy clinician as well as the specialist about what genetic factors play a role in particular disease, and is a test available, and if so, where can you have it done? I should say, by reason of parity, that there are other websites as well that are extremely valuable for surveying the genome, and I think experienced researchers tend to learn how to use each of the three major genome portals for particular purposes. This one, the one operated by our good colleagues at EBI and the Wellcome Trust and the Sanger Institute, the Ensemble site, also has a somewhat different view of various species. They've been particularly involved in the recent work on the mouse. There is now a wonderful draft sequence of the mouse that is far more contiguous and assembled than most people expected, freely accessible from the black 6 strain. And you can see that both at Ensemble and at NCBI and at this site from Santa Cruz. This particular browser, built by a graduate student named Jim Kent, is also very popular amongst postdocs in Building 50 that I talk to from time to time because of the way that it's constructed and the view that it gives you various features of the genome. It has some overlapping features with what the map viewer from NCBI will offer you, and some things are done a little differently, and it would probably be a good thing to do a comparison for your own question and see what is giving you most easily what you need. This is just a quick screenshot from the MET oncogene region of chromosome 7, showing you the kinds of features that appear on the various tracks, which you can turn on or off at will. So there are a lot of resources out there to allow the investigator to get access freely and immediately to the data. And yet I think it is still true that many people are having some trouble figuring out how to use these tools. And in that regard, we also try to offer some help here through our own website, the NHGRI site, which has just undergone a major revision. And our new site just went up about 3 weeks ago. And we managed to get the URL called genome.gov. So if you forget NHGRI, you can quickly find us, and this will get you to a whole lot of information about genomics, including online resources, which has a bullet up there which will get you to many other data sets besides the ones I mentioned, as well as a bunch of timely announcements, like, for instance, this one down here in the corner, which you're now part of. This is the site as it appeared this morning. I guess by tomorrow it'll have to say something else. I would also point you to a tutorial which a number of us have been involved in, particularly Andy Baxavanis and Tara Wolfsberg, putting together a set of examples of the kinds of queries that biologists and medical researchers would be likely to want to ask of the databases And this is going to be in a printed form, distributed with Nature Genetics, but it'll also be available for free on the web, and you can even send in a card and get your own hard copy. And this is really not intended so much for the expert as for perhaps the graduate student in a physiology lab who's having a little trouble figuring out how to get access to all this information and doesn't have a bioinformatics expert at their elbow to help them. If you go through the examples in this tutorial, which include screenshots of what you would see if you posed a particular query, you will learn most of the major features of the NCBI, Ensemble, and Santa Cruz sites in a fashion that I think will be very useful. So watch for this. It's coming in about 6 weeks. I might say for medical librarians who are often put in the position of trying to teach people how to use these tools, I think this might be a very valuable resource for that purpose as well. Well, that's a bit of a romp through some of the data and how to find it. Let me move now to the question of where we go from here, because some might have argued that the Genome Project was about getting the human sequence, and that's just about done, so maybe we ought to just close down and say we did it and move on. Well, in some ways that's true, but in other ways what we're talking about here is the end of the beginning. We will, by the way, finish the human sequence, which still is in draft form, by next April. In April 2003, we'll see the coincidence of the 50th anniversary of the double helix and the completion of all the human chromosomes to the same high standards of accuracy that have now been achieved for chromosomes 20, 21, and 22. So we are working very hard with all of the 16 centers involved in doing that finishing to be sure that that course is maintained, and I can tell you that as of last week's discussions, we're very much on track to do that. We'll be meeting again in a month in Japan to survey the situation. But in fact, most of the chromosomes are already in remarkably advanced shape. And I think when April comes around, you will see a really wonderful genome sequence for the ages. Now, I should say as one caveat, that will not include the complete sequence of all the human centromeres. The centromeres are these long repeats of alpha satellite sequences, which are unstable in any cloning vector that we know of, and therefore cannot really be recovered in their native form. Until we come up with some other technology that allows the sequencing of those components, they will remain somewhat ambiguous. The good news is there are probably very few genes in that region. The bad news is, of course, centromeres are pretty important, although it does seem as if the primary function of the centromeres is the fact that they have these very long repeats, and there may not be nuggets of surprising sequences buried within the repeats still waiting to be uncovered. So with that caveat, I think one can think of April as the point at which the human genome sequence is essentially complete. But we do have all these other things we have to do, and have to do urgently if we want to understand this and apply it for medical benefit. We need to understand how the genome works. So things like microarrays, which are being explored by numerous laboratories around the world to understand how genes turn on and off, are a vigorous part of the new phase of genomics. The effort to look at proteins in a systematic way instead of one at a time, so-called proteomics, is really just getting going and has many technical challenges to face before one could say we're really ready to characterize the whole set of human proteins at the same level that we have tried to do with DNA. But it's a worthy goal, and one which NIH is going to be very invested in over the course of the coming years. We believe that the comparison between genomes, telling you, in fact, which part of the DNA has been most strongly selected by evolution, is one of the most powerful ways to identify which are the functional elements, the parts that we would most like to understand. And so we are now invested in sequencing quite a long list of genomes, with the mouse and the rat and the zebrafish already well underway, and in the case of the mouse already moved into an advanced assembly. Many other organisms are about to get started. The chimpanzee and the chicken, for instance, will be started this year, and a number of others are waiting in the wings. We have a process where people who are interested, or communities who are interested in a particular genome being sequenced, can apply to have that done. These are reviewed then by a panel that tries to decide what is the most valuable genome for information amongst those that have been proposed, and then we prioritize those and put them into the pipeline of our sequencing centers as the capacity becomes available. And that is turning out to be a very interesting process in its own right. That panel will meet again in about a month, and there's going to be a big argument between the cow people and the dog people about exactly which of those two mammals ought to be the next one in line, because there are strong proponents for both. And I get phone calls both from the Bovine Breeders Association and the American Kennel Club, and you can imagine which is proposing which. But I think it is a good thing that our sequencing capacity now in the public genome centers is really quite prodigious, and the costs have come down dramatically so that we can afford to do quite a lot of this for a manageable cost. And when you look at what comes out of it in terms of the comparisons, particularly with some of the new software methods that are being developed to mine the maximum amount of information out of these comparisons, we are going to be very glad that we have as much of this data as we can generate. The medical genomics part of this is focused particularly on trying to understand how it is that various people are predisposed to various illnesses. And that means that we need to study intensively this 0.1% of the genome that varies from person to person. And in that regard, there is now a very vigorous effort as a public-private partnership to build a catalog of those roughly 10 million places in the genome where there is a common variant in our species. And when I say common, I mean a variant where the less common allele at this single nucleotide polymorphism is perhaps 1% or greater. The rarer ones, of course, there are almost infinite numbers. There are probably a variant in every base pair in the genome somewhere in somebody in the world, unless it happens to be a dominant lethal. But the ones that we want to particularly catalog are the ones that are a bit more common, because the expectation is that within those common variants will lie the clues to hereditary factors in diabetes and heart disease and mental illness and virtually every disease that you can name. And so the development of that catalog, and particularly figuring out how these variants relate to their neighbors, is a very high priority. If this is a typical page, this comes from chromosome 7 of the instruction book, this might be your sequence, and this might be mine. There are 2,000 base pairs here. There are 2 variants that are already determined and sitting in the DB-SNP database that NCBI operates. Those 2 variants might very well not matter, but one of them might actually be one that changes an amino acid or a regulatory signal and predisposes to a disease. What we've recently realized is that to do that kind of case control analysis that you'd like to do with these variants to figure out which ones are associated with a particular illness, we may not have to go and sample all 10 million of them in order to get that answer, because they are correlated. So if you have a C there, it's very likely that you have a T there. They are not going to be independent of each other. This is because the genome essentially is broken down into what we call haplotypes, that is, segments that are in very close association with each other. And that means that if you're doing this kind of association study, you need not sample all 10 million of the variants. If you sample a carefully chosen set of perhaps 250,000 of these, you should be able to essentially view all of the major variation in anybody's genome and identify the segment that's associated with illness. And that would then allow you, if you were sampling for, say, the purple versus the green haplotype in gene A, or the orange versus the blue haplotype in gene B, to identify a segment that's associated with disease. In this case, gene A doesn't look as if there's anything going on here, whereas in gene B, the orange haplotype seems to be conferring risk, and would then very much make you want to go in and see exactly what variant in there is associated with some biological consequence that would account for this outcome. I think one can imagine that those sorts of association studies are going to be happening quite vigorously. The haplotype map project that will allow us to pick out the right variants to score is about to get started. We'll start in about October, and will probably occupy about a 2-year time period, funded by most of the NIH institutes in association with the private sector, and particularly with several countries, including Japan, China, the United Kingdom, and Canada. And with that haplotype map and carefully constructed case control studies, most people would project, and I will too, that over the next 10 years, and that may be actually rather conservative, we should find the major contributing genes for common diseases, such as the ones noted on this list, and others as well. So that will be pretty exciting. Those will, each time you find one of those, point you towards a pathway that is involved in disease pathogenesis. And it does so in a fashion that depends not at all on what we already knew about the biology of the disease. And that's good, because if you ask very penetrating questions about what do we really know about the causes of diabetes or mental illness or even cancer, it is often the case that we are fairly ignorant. And the beauty of the genome approach, as will be manifested in this particular haplotype map effort, is that it is a complete approach to the whole instruction book. You don't have to have a hunch about where the answer is going to be. If it's there, you're going to find it. That is what genomics offers you. You look at the bounded set of information that constitutes the human genome, and somewhere in there you're going to find the answer you're looking for. Well, how then, to move on to medicine, will this affect the way that medicine is practiced over the course of the coming years? What's going to be this impact, and how soon is it going to come? In making such predictions, I am always somewhat mindful of the fact that people who try to make such predictions are often woefully mistaken in the estimates they make of how long things will take to come to pass, or even if they're going to come to pass at all. And yet at the same time, I think people are interested in some speculation about where this is going in order to prepare for it. And we are probably obligated as researchers, as physicians, and as members of society to try to do some of that forecasting so that we aren't caught flat-footed when something comes along, and we really have done the thinking about what we need to do to get ready for that. So I will try to make some of these projections, recognizing that people who have preceded me, some of whom are quite distinguished, have made some real howlers over the course of time. Well, for example, here is a prediction from somebody who was quite an expert on the topic of computers. I think there's a world market for maybe 5 of them, he said. That was Thomas Watson, who, of course, was the chairman of IBM. Well, it was 1943, okay, so that's like prehistoric. But, you know, that's sobering that somebody would have said that within the last 100 years. Another prediction, this one from a professor. You know, professors occasionally get things wrong, too, I hear. This was a comment written on a term paper written by a guy named Fred Smith, who happened to be at the time a student in a business class at Yale University. And his term paper, the assignment was to propose a possible business plan and then defend it. And obviously the professor wasn't so impressed. Well, the student went on to actually follow up on his plan, and after he got out of Yale, actually went out and started such a company. The company was called FedEx. So I guess Yale doesn't always have the answers. Another prediction in a different field. This is from a evaluator of music. We don't like their sound, and guitar music is on the way out. Decca Records rejecting the Beatles in 1962, and no doubt regretting it somewhat later. And particularly for you computational gurus, here's my favorite of all of these. 640K ought to be enough for anybody. And you may guess who said that. Yes, it was Bill Gates. And it wasn't so long ago. So as I try to now make predictions over 10 or 20 years, keep in mind that other very smart people have stubbed their toe here. Although you will notice that all of these predictions were wrong because they were insufficiently sort of optimistic about the future. Maybe mine are overly optimistic. You can decide. Well, where is this all going to go in terms of medical application? This diagram, which I'll show you a couple of times, is a way of organizing over the course of time, which is over here on the y-axis, what kinds of steps are likely to happen as genomics gets applied to disease after disease. The steps that I've largely been talking about in terms of the genome and the map of human variation will allow us to get to the point of finding the genes that underlie hereditary predisposition to virtually all illnesses over the course of the next 5 or 10 years. And some of them, of course, are already very much in hand. Now, what happens then? Well, of course, you keep moving down the diagram. The clinical implications start here, and the first one you come to is diagnostics. So if you do identify what the risk factors are for something like colon cancer, you then have the opportunity to figure out who is at highest risk, and in a situation like that disease, offer them something in the way of preventive medicine, like intense medical surveillance to pick up a polyp while it's still easily treated. In fact, that is already possible to do in certain instances with diseases like colon cancer. Here's a pedigree, for instance, where you see 3 individuals affected by colon and uterine cancer, a pedigree which would tip off a medical geneticist or an oncologist who is well trained in genetics to strongly suspect that this family might be harboring a mutation in one of the DNA mismatch repair genes, particularly, for instance, MLH1 or MSH2. And in fact, this is a family that went through a research protocol and had the analysis done, and it turns out the proband, in this case this person, does in fact have a mutation in MLH1, which probably accounts for his illness and that of his mother and his sister. And of course, there are other people now in this family who are very interested in this finding and want to know what their situation is. These two individuals, this woman, this daughter, this man here, are all at 50% risk of having inherited that same mutation. If you have that mutation, your risk of getting colon cancer is about 60%. If you're a woman, your risk of getting uterine cancer is also substantially elevated. But for both of those, we do have pretty good medical surveillance approaches, which have been shown in clinical trials in this kind of family to be life-prolonging. And so this is a circumstance already where diagnosis can be quite valuable. This is a family that decided to take advantage of that, and these three individuals turn out to be mutation carriers. They are all now in colonoscopy programs, some of them starting as young as age 35, and having those done quite faithfully. And already a polyp has been picked up in one of these people, a polyp that might very well have gone on to be a metastatic malignancy over the course of time. This is not the disease familial polyposis, which is much more dramatic, where there are thousands of polyps in the colon. This is so-called hereditary non-polyposis colon cancer, which is much more common. Probably 1 in 400 or 500 people have this condition. And yet, the opportunity for intervention here is really very compelling, because while there are relatively few polyps in these individuals, their malignant potential is very high. While we would like to see this same kind of possibility applied for a long list of diseases, I think it's fair to say at the present time the instances where testing and preventive medicine is available is a fairly modest list. But that list is going to grow, and it's going to grow as things come down the pipeline here from the use of the genome to identify predisposition genes. Another aspect of diagnostics is this business of pharmacogenomics, wherein you use the test not to predict who's going to fall ill, but who is going to have a bad reaction or a good reaction to the drug that they're given when they fall ill. And this is a field which basically didn't get much attention at all until about 3 or 4 years ago, and now there are several journals devoted to the topic and a raft of papers that are coming out reporting how people's ability to respond to a particular drug varies as a consequence of their DNA sequence. Maybe the most dramatic example, and one which is now part of the standard of care in pediatric oncology, is the use of 6-mercaptopurine in the treatment of acute lymphoblastic leukemia. This is part of the standard therapy, a very successful therapy that now cures something like 95% of children with this previously fatal disease. But there is a terribly sad and wrenching consequence where about 1 in 300 children, having been given the appropriate drugs at the appropriate doses, will develop a very severe toxic and sometimes fatal reaction to 6-mercaptopurine. Well, it was reasoned that there might be an explanation for that at the genetic level, and investigators in several places were in fact able to identify the cause, and that is that the gene that encodes an enzyme called TPMT, which is involved in metabolizing 6-mercaptopurine, carries a null allele, which is present in about 1 in 10 people, which means that about 1 in 300 are homozygous for that, and as a consequence are very unable to metabolize this drug, even though they otherwise seem unaffected by the absence of that enzyme, or the virtual absence. So now knowing that, one can test for this ahead of time before administering the drug, and that is part of the standard of care. And if you identify that that child is that 1 in 300, you can still give the drug, but you give it at a greatly reduced dose. They then still experience the benefit of the drug in terms of inducing remission without the side effects. And this is but one example of what is a very rapidly enlarging set of data sets that suggest that pharmacogenomics is not far away from becoming part of the standard of care for many conditions, including things like asthma and heart disease. So don't be surprised if sometime in the next 5 years, when you are at your physician's office and they are ready to write you a prescription, if they say, you know, first we better do a genotype and be sure this is the right drug for you. This could be a very good thing. We all know from reading the literature and from various reports, particularly a recent one from the IOM, that adverse reactions to drugs are a profound cause of morbidity and even mortality in our medical system. And the expectation is that not all of those, but a substantial fraction, may well be predictable if we understood exactly what goes on in variations in human metabolism and were able to make this kind of prediction and avoid using the wrong drug in certain circumstances for somebody who's not going to be able to tolerate it. Of course, everybody would like to believe that the diagnostics and the preventive medicine will keep us all healthy, but we will still fall through the safety net and we will fall ill, and we hope that what will be waiting for us there is a new kind of therapy, perhaps more successful in many circumstances than what we currently have available. Because except for some conditions like infectious diseases and a few other things, most of the diseases that we currently treat, I think if we're honest with ourselves, our treatments are not really all that satisfactory. And it often is the case that they were arrived at relatively empirically. They do have benefit, but the benefit is not as good as we'd like, and the side effects are often much less pleasant than one would hope for. So we all dream of a time where our understanding of biology is much more precise, and so we can therefore institute therapies that are based on rational design. Some of those may be a case where the gene itself is used as the therapy, which is what gene therapy is all about, and that after a very bumpy course over the last 10 years or so, especially with the death of Jesse Gelsinger a couple of years ago, now seems to be making some steady but limited progress in areas such as inherited immune deficiency disorders and hemophilia. So I do think gene therapy will find an important niche in the armamentarium of medicine, but I suspect it will be limited to a certain set of conditions. I actually think the major benefit of genomics for therapeutics is going to happen by moving down this pathway, where the information about the gene allows you to design a therapy that is much more precisely targeted to the problem than what we've previously had available. And that is perhaps best exemplified as one that's already made it down here to the bottom of the diagram by the drug Gleevec, which is approved by the FDA just about a year ago for the treatment of chronic myeloid leukemia. It comes about, this particular type of leukemia, because of a chromosomal translocation that produces a fusion protein between the genes called BCR and ABL. That protein transforms white cells into malignant white cells, resulting in this kind of a blood smear. Brian Drucker, who spoke here at NIH about a month ago, and his colleagues at Novartis reasoned that if they could design a drug that blocked the active site of that protein, which binds ATP normally and acts as a kinase, they might be able to block this cascade of events. And sure enough, this drug has been dramatically successful in inducing remission in most patients with this previously rather untreatable disease, and gives us all a lot of hope that this paradigm is going to be successful for other diseases if we could just understand them as well at the molecular level as we understand CML, where there have been 30 or 40 years of hard work to figure out exactly what's going on. So I think it's fair to say that there's a lot of reason to be optimistic here, that these advances, both diagnostics, preventive medicine, and therapeutics, are going to come along over the course of the next 10, 20, 25 years. But at the same time, people are concerned about whether those same advances are going to be treated appropriately in terms of providing protections against misuse and an opportunity for societal dialogue about what boundaries ought to be applied, if any, to the application of genetics when it goes beyond the medical arena. In this regard, Jim Watson, my predecessor, I think very wisely decided that a fraction of the budget of the Genome Project, about 5%, would be devoted to studying the ethical, legal, and social implications. And so the ELSI program was born and has been, over these last 12 years, the largest investment in research in bioethics ever mounted on any topic. Out of that has come a cohort of investigators, some lawyers, some ethicists, some social scientists, some theologians, who have considered what the major challenges might be from this rapidly accelerating pace of genetic information, and then have proposed possible solutions to limit the likelihood of trouble. And I think we can say this has been, in that regard, a very successful experiment. We have a wonderful wealth of literature and investigators who are expert in this arena. What has been more difficult is to figure out how, in some instances, to implement those solutions, because that involves the political process. And so, even though scholars have concluded from many different perspectives over the course of these years that what we really need to prevent genetic discrimination in health insurance and in the workplace is effective federal legislation, we do not yet have that. The good news is that we do have in the Senate of the United States this year a very effective worded bill from the Democrats and a similarly effective worded bill from the Republicans. There are some differences in the definitions and the penalties, which we are hoping they are working out this summer. And the President has spoken very strongly out in favor of the need to pass such legislation. The problem is, though, that our process seems to operate most effectively when there is a crisis. And the idea of doing this in a preventive way, to anticipate a problem that is largely in the future and not so much in the present, since most people have not yet had a genetic test, is not something that our system responds to as well as I wish it would. And so it will take continued determination on the part of effective leaders in order to push this across the finish line. But if we don't do that, because we all are flawed after all, this is about all of us, increasingly we'll have the chance to find that out. And if we don't have the protections, then genetic discrimination will, as the cartoon indicates here, cause us to all get screwed. So be prepared. If we don't take action here, the consequences are not just for a few unfortunate people, they could be potentially for all of us. We have a big educational challenge. If we are going to mainstream genetic medicine, then the providers, many of whom have had no exposure to this discipline, are going to need to acquire the skills of a genetic counselor and are going to need to understand some of the intricacies of genetic tests that are not giving you yes or no answers, but are giving you statistical statements about relative risks. And we have quite a challenge here, I think, to prepare people for that. One possible solution to that is this organization called the National Coalition for Health Professional Education in Genetics, or NCHPEG, which, as the slide will tell you, has now enlisted the help of quite a number of professional societies and has a bold agenda based upon various core competencies and other efforts of how to achieve this sort of literacy over the course of the next 3 to 4 years. But this is going to be a huge undertaking, and it will not just be physicians, but also other healthcare providers, especially nurses, who need to acquire these skills. I think we have to continually be concerned about access questions, given that medical treatments are not, at the present time, equitably distributed in this country, and the fact that new technologies tend to be even less equitably distributed. Are we facing an outcome where these advances in genomic medicine will find themselves into the hands only of those with the highest levels of financial worth and education? And that would be a very unfortunate outcome, and yet one which at the present time one doesn't see an easy solution for, given the way in which our current dialogue about healthcare has stalled. What about the issue of studying variation and its effect on the always contentious question of race and ethnicity? There's a lot of good news here from the science that ought to be useful in this dialogue, in that this 0.1% of variation that we know about is actually variation that mostly pre-existed in our common founder pool of about 10,000 individuals who lived in Africa 100,000 years ago. And there is therefore no scientific reason to draw any precise boundaries around particular groups and say they're different. We are, as a species, incredibly alike, much more so than most other species on the planet. And science will not support a prejudicial argument about the superiority of any particular group over another. But I'm not sure that that is necessarily being received in a welcoming way by those who are determined to make war with each other, as exemplified by this particular cartoon, where these two individuals are apparently not pleased with the revelation that's being offered up about the similarity of their DNA sequences. And this cartoon is sort of funny and sort of sad because, in fact, when you consider that in this instance, particularly if you go back and read the Old Testament, these two individuals are actually probably traceable quite dramatically by history back to a common ancestor, and yet somehow they and the members of their respective groups imagine that they are profoundly different and share almost nothing in common. Perhaps science can help in a small way here, but I'm not so naive as to believe that it's going to turn the tide of all of these other animosities that we seem to have developed between groups who identify themselves as somehow different than others. Where will we set the boundaries, or will we at all? Is there a limit to the application of genetic technology when it goes beyond the treatment of terrible diseases and on to the point of trying to enhance various traits? Most of the scenarios that are put forward here in movies like Gattaca, for instance, if you happen to see that, are pretty unrealistic scientifically because they ascribe to DNA a much more powerful role in various features like behavior and intelligence and athletic ability than actually DNA deserves, because most of those features are ones that are heavily influenced by environment, not to mention by just simple choices that we make about exactly what to do with our lives. Nonetheless, I think there probably will be issues that society is going to have to wrestle with about whether or not we want to apply genetic technologies in a completely unrestrained form to try to skew the odds in terms of the characteristics of future generations, or whether we think that ought to be off limits. That plays into this final question, which is more a philosophical one. Will we succumb to genetic determinism by getting so excited about the DNA molecule and what it has to offer that we will begin to neglect the role of the environment, which, as I said at the beginning, is a critical player in most of the common illnesses that we would like to understand better, and certainly a critical player in many of the non-medical aspects as well. And will we also, in the process of focusing on DNA, begin to think of ourselves in a more mechanical way, undercutting the value of the human spirit and its power, and even our need for God? And that would be a truly tragic outcome from a scientific effort which aims to explore the mechanical side of human biology. To conclude that that's the only side there is would be a fundamental error. In that regard, I'm sort of fond of this cartoon because for a while I was referring to the genome as the book of life, and others were as well, and perhaps this is a bit of a reminder that there's a bit more here than just that when you're talking about life itself. So let me conclude by saying I think we have a lot of challenges that lie ahead, but one could hardly imagine a more exciting time to be working in science, to be working in information science. It is a time where, as a team, we're going to have to tackle problems together, much more so than perhaps in the past, where single investigators might be able, in a lonely way, to make a major advance. I think in this era, much more of the major advances will happen by teams that involve multiple different expertises. But what an amazing opportunity lies in front of us to really unravel how human biology works and to apply that for good purposes to decrease human suffering and to advance the human cause. I'll leave you with a final quotation, which is not actually from a scientist, not actually from a philosopher, but from an athlete. And it's my favorite quotation because it, in a very few short words, provides a recommendation about how to prepare for the future. This quotation comes from the great one, Wayne Gretzky. And after one particular game where he had scored I don't know how many goals and nobody else had scored any, the press was asking him, how is it that you always seem to be a little better than the rest of the guys on the ice? And he said, it's a very simple principle. I skate where the puck is going to be. Well, okay, that sounds like a good charge. Let's all go out there and skate where the puck is going to be. Thank you all very much.
Dr. Collins, thank you so much for this enlightening and entertaining and instructive talk. To those in the audience, we have time for your questions, and I only ask that you look in front of you and find a little button that will activate the microphone and put a light on so that the others can hear you and in the spillover space can hear your questions. And you choose which of these lights you point at.
Questions? In the back, somebody who doesn't have a light, but I can repeat the question. So the question is whether I have a position on re-engineering of the human germline, recognizing that some popular books, the one referred to by the questioner is Lee Silver's book called Remaking Eden, have proposed this as an opportunity to engineer our species in new and interesting and, in his view, inevitable ways. I have a very different view. I think that altering the human germline will be, for the foreseeable future, an unethical activity on the sheer grounds of not knowing its consequence. If we are contemplating going in and changing the fundamental genome in a fashion that's going to get passed on to future generations, which is what germline alteration would have to mean, then you would have to know with great certainty that what you are doing is not going to have unexpected and untoward consequences. Until such time that we have a foolproof way of going in and fixing one nucleotide without altering anything else in the whole genome at the same time, it is hard to see how one could argue that this is a process that you could do with complete safety. Now, some people have argued, well, you know, you put a suicide gene in there and you pop the thing out again, or you use an artificial chromosome. Yeah, okay, those are options, but they do not rise to the level, even close to the level, of something that one could say confidently is associated with safety. And when you consider that such human experimentation would be placing at risk not only the individual receiving the therapy, but all of their future offspring, this does not seem to me to be the kind of experiment that an ethics board could justifiably accept. Finally, on top of that, for medical purposes, it is very hard for me to come up with scenarios where germline gene therapy is really necessary, in the sense that most of the conditions that you might want to treat with some kind of genetic intervention could be dealt with by much simpler and safer strategies that don't require us to develop all of this other technology that we're a long way from having. Here's a light. Yes, ma'am. Oh, keeps going on. Can you push it on again?
As you wisely pointed out, the major advances that are expected of human genome is in treatment of human disease, and more specifically in cancer. Can you say that today we can take the advances that were produced in multiple studies of microarray and genomics in diseases such as breast cancer, and are we ready to proceed with some modification of therapy for this disease?
So I had a little trouble hearing the question, but I think you're asking whether we could say now that genomics has provided advances in the treatment of diseases such as breast cancer. Am I mostly right there? I think it's still early days to point to a long list of examples that have already gone all the way through the process and are now considered to be standard of care and where you can trace a direct connection to genomics. I guess the Gleevec example is perhaps a good one. In breast cancer, I think one can look at what we've been able to do in terms of Herceptin, which of course is based upon a genetic understanding of changes in the tumor, to say that there is a real positive consequence there of understanding at the genetic level what's happened in that cancer. There was a very nice paper with regard to microarrays in breast cancer a few months ago that showed that with a carefully chosen set of genes whose regulation one could study, you could make a very good prediction in women who have a tumor under 5 centimeters, who are under 55 years of age, and whose nodes are negative, which of those women are going to have recurrence and which are not, which would be incredibly valuable to know because it would allow you to choose which people should get adjuvant chemotherapy and which people don't need it. And that particular study, I gather, is already leading to an implementation of that as part of the evaluation of a breast cancer in some centers in Europe, and I think probably we're not far away from seeing that happen here as well. So while I don't want to overstate the immediacy of these consequences, I do think cancer is the place where they will come the most quickly. And I think they are not far off for many cancers in, say, the next 3 to 5 years, both in terms of improving diagnosis and choice of therapy and in terms of coming up with brand new therapies. I understand there are over 100 drugs now in clinical trials for various types of cancer that are based upon an understanding of the oncogenes or tumor suppressors or other cancer genes that are involved in particular types of tumors. Very much in the way that Gleevec was designed, these drugs have been designed either to knock out the action of an oncogene or compensate for the absence of a tumor suppressor. Whether those 100 or more drugs are going to yield some more home runs like Gleevec, I don't know, but I sure hope so with that number, and I expect there probably will be some. And some of the noises one hears suggest that may not be that far off.
Yes, could you comment on the possibility of the effects of eating genetically modified produce on humans in the future?
A nice, simple, non-controversial topic. Well, I confess I am not an expert on genetically modified foods. I've certainly read some of the literature, and I've read a lot of the press debates and been aware of a lot of the public concerns. I think to summarize it all, I think there is very little compelling evidence that there are risks associated of a real sort with genetically modified foods as they are currently being propagated. At the same time, I think it's also safe to say that the amount of data that's been generated on safety is probably a lot less than we now wish we had in order to try to make that case more compellingly. And perhaps an opportunity was lost a decade ago to collect more of that data. And now I think the public is sufficiently stirred up, particularly in Europe, that it makes it very difficult to provide assurances that this is all actually safe without being able to point to extensively large studies to document that, even though that does seem like the most likely conclusion. I think there is a bit of a lesson there, and that perhaps is one that Jim Watson wisely realized. And perhaps if Jim Watson had been running genetically modified foods, things might have turned out differently. Namely that you don't want to wait until you're 10 years into a project to try to begin to think about what the consequences might be and how the public might react to it. And we're in a bit of a mess there with genetically modified foods. Having said that, as somebody who spends a fair amount of my time also worrying about the health of the third world, without genetically modified foods and the various strains that are being created to improve crop production, we are facing not just a hypothetical possibility of a few people potentially having a bad reaction to some gene that's been spliced into corn or rice, but millions of people dying of famine. And in the discussions about genetically modified food, that should certainly be front and center. And those who wish to say that this is a bad thing and we shouldn't do any more of it should realize what the consequences are of what they're proposing. Yes, in the back.
For graduate students or doctors?
So the question is, from a pre-medical student, or a medical student, I'm sorry, what are the possible venues for specializing in various types of genetic research, such as genetic therapy, that would allow doctors to play a significant role in this? I think there are going to be lots of those possibilities. Virtually every area of research is now incorporating genetics and genomics into the mainstream of their approach to disease. There are certainly some areas that are particularly focused in that regard. My own training is as a medical geneticist. I was trained first as an internist and then did a fellowship in this board certifiable specialty called medical genetics, which allows one to focus very precisely on illnesses that have a strong hereditary component and on research approaches to fixing those problems. But one can do this as a research activity from many other approaches in medicine and pediatrics, virtually from whatever platform appeals to you. In fact, my hope would be that people will follow their clinical instincts and then incorporate into that field the genomic attitude so that we won't see this only spilling out into some parts of medicine, but into all of it. This is going to become sort of the central way that we approach problems in research, and before long, I think it will also become a very central way in which we treat patients in the clinic. Yes?
When you say that next April a final completed genome will be printed, is that going to be like a conglomerate basically, or an averaging of many different genetic makeups, or what exactly is that going to consist of?
Yeah, whose genome is this anyway? Well, the public project worried about this and felt that it would be important to get DNA from individuals whose identity would remain anonymous, because would you want your DNA sequence up on the internet, given all of the things that people might decide to do with it in terms of taking away your health care? At least right now, it didn't seem like a safe thing to subject anybody to who could be tracked down. So we put an ad in a newspaper in upper New York State, which happens to be where the lab is that we're making the libraries that the Genome Project was primarily using, and invited volunteers to come in and give a blood sample. And about 100 of them turned up, and they were diverse in their ethnicity and their gender. And all of those people, except one, I think, after hearing about what the project was about, decided to participate, signed consent, gave a blood sample, and went home. We made DNA from all of those samples, and then we took all the labels off. And we took only about 5 of those that were used actually for the majority of the sequencing. But we don't know which 5 they were, and they don't either. And they were told that there was a high likelihood that their sample wouldn't even be used for their own protection. So what that means is that the public sequence is a hodgepodge of a series of individuals' DNA. I'll tell you actually about 60% of it comes from one particular individual, because that library was characterized particularly in depth, and so it was one that a lot of people wanted to use. It doesn't matter a lot from the perspective of the fact that 99.9% of the answer would be the same. What does matter, of course, is that 0.1%, but the sequence itself is not the place to sort of look at for that. This is another part of the project, which is characterizing variation. In that regard, we have 450 DNA samples that are roughly evenly divided between Europe, Asia, Africa, and Native Americans. And those are the samples that are being used to identify the roughly 10 million variations in the genome, which are all then mapped back across the sequence. So you ultimately get both things. You get the stuff that's the same, and you get the stuff that's different, depending upon a rather different set of DNA samples to get there. The sequence that you can derive in the subscription database from Celera, you may have heard, has a somewhat more identifiable source, in that originally this was intended to also be anonymous, but more recently Dr. Venter admitted, actually rather proudly, that more than half of that sequence derived from himself. And you can decide what you want to do with that information. Other questions? Yes?
I have two questions.
Oh.
The first is, what can we as scientists do to prevent genetic discrimination from taking root and becoming really prolific?
Mm-hmm. So the first question is, what can scientists do to prevent genetic discrimination from taking root and proliferating? Well, I think we're pretty close to seeing, at least in the U.S., an effective solution, but it hasn't quite happened. Scientists have a role to play. I think the day where scientists go in the lab, close the door, and let somebody else worry about the consequences of our research, that day is past. And I think all of us carry a responsibility to be involved in these discussions about policy and contributing to the public debate, because we possess information that other people are hungry for. We don't necessarily understand how the government works better than a lot of other people. We don't necessarily have the corner on the market on what is ethical behavior, but we do understand how science works. And we should be, I think, volunteering ourselves to be part of those discussions and looking at the recommendations that have been made and talking to our elected officials about what they need to do. Now, me as a member of the executive branch, I am prohibited from lobbying, so I can't call up a senator and say, you should do something about genetic discrimination. But if I get asked, I can answer, and so I'm always looking forward to being asked. And increasingly, the Congress is asking. But it isn't going to happen. Nothing happens in our political process unless there is a groundswell of noise saying, this is important. Don't put this one off until next year or the year after. Do it now. What's your second question?
Well, I've been watching you over the last 2 years. I'm a member of SACRIS, and so I saw you speak there. And so now that the project is basically done, what are you going to do with your time?
So now the question is, now that the project is done, what am I going to do with my time? Well, I haven't discovered that there's a lot of extra time. A couple of people from my lab are here, and I think they would tell you they don't see me much more than they used to, maybe even less, it seems like, because actually it seems to me that what we were doing before was the straightforward part, the part that was pretty well defined, working out those 3 billion letters. What we're into now is this long list of very exciting follow-up projects, studying all these other genomes, building this catalog of variation and figuring out how it connects with disease susceptibility, figuring out how the genome functions, getting into proteomics and developing all the technologies to do that. So my plate is more full than it ever has been, and I'm glad for that. This is a very exciting time, and it's a wonderful privilege to have a chance to be here at NIH leading this particular effort, working with some of the smartest people on the planet, all around a goal that I think really has a certain nobility to it. We're doing something important here, something that should actually change things for the better, as long as we pay attention to getting the science done right and also making sure that we put the appropriate protections in place so it doesn't get misused. And that, by the way, is another major part of how I spend my time, is all those ethical, legal, and social issues. If we don't pay just as much attention to those as we do to the basic science, then we may find that this revolution has a lot of casualties. And we can prevent that, but we have to be thoughtful and thinking ahead about how to make that outcome a good one. That seems like a good note to end on, and I don't see any other. Oh, well, there's one more up there. Okay, yes?
It seems to be particularly important to make a distinction between trait and disease in the context of genetic testing. Can you just briefly comment on if you can somewhat draw a line between disease and non-disease? I know we have a limited amount of time, but...
So that's a great question. The question is, if there is a reason to try to treat genetic testing somewhat differently, if you're talking about disease versus trait, how do you draw the line between them? Is there a possibility there of having that be very precise? This is a really hard problem because, in fact, the line is not going to be precise. I mean, there are certainly extreme examples. Well, not even extreme examples. There are lots of examples where it's pretty clear which one you're talking about. If you're talking about, for instance, gender, well, that's a trait. That's not a disease. If you're talking about Tay-Sachs disease, that's a disease. That's not a trait. But what about, say, obesity? There's a circumstance where we know there are strong genetic contributors. We certainly know that morbid obesity is morbid. That is, that it causes untold havoc in terms of medical consequences. And if we could figure out ways to prevent those kinds of medical consequences in those who are most struggling with significant obesity, well, that would be a good thing. But would the application, if we figured out what genes are involved in this, of offering to well-heeled couples the chance of choosing an embryo that's more likely to be svelte than chubby, is that a good thing? That really challenges, I think, the notion of disease, and it sounds much more like an application to traits, and especially when you're talking about selection for the next generation, I think it makes most of us pretty darned uneasy. These will not be easy lines to draw, and I think the only way we can proceed is to do the best we can, and to be sure that in doing the best we can, we've heard from a lot of different constituencies. I might point out as one final comment that all of these issues about traits and diseases and the effort to try to do something about them are received with very mixed responses from the disability community. If you're somebody who's involved in some kind of disability and people are talking about, well, wouldn't it be good if that didn't happen anymore? What does that say? We have to be very careful here about the way in which this creates a mindset that the only thing that's acceptable is the absolutely perfect specimen. We're never going to get to that. And furthermore, the notion that we should get to that is in some subtle way a way of saying that people with disabilities are less valuable than others. And we have to be very careful in that particular perspective. And we have a fair amount of the ethical, legal, and social debate that has been focused on that issue, not with entire success, I might say, because I think there's still lots of consequences of this that need to be worked through and more broadly understood, not just within a small group of ethicists and philosophers, but by society at large. Well, thank you all. It's a wonderful set of questions. This has been a great discussion.
Thank you, Dr. Collins. It proves that there are still heroes amongst us. And the audience did ask wonderful questions. And as a reward, there is a reception outside. Please join us. That was terrific.
Oh, thanks, Don. Thanks for the chance to talk to you.